This is Ocular Disease and Refractive Error Spotlight, Retinitis Pigmentosa. Retinitis pigmentosa is a group of rare genetic disorders that involve a breakdown and loss of cells in the retina. Approximately 1 in 4,000 people in the United States have it, and it's an inherited disorder of more than 50 genes. Symptoms include rods that are more severely affected than cones, night blindness, and progressive loss of the visual field. Ultimately, retinitis pigmentosa is due to mutation in the rhodopsin genes, and this is the primary photopigment of rods. Due to the rods being primarily affected, all the damage is in the periphery and not the fovea. Here we have two OCT images. On the left, one with retinitis pigmentosa, and one in the middle, a normal retina. If we actually look closer at them, the central macular structure of this OCT image is relatively preserved in either image. The patients with retinitis pigmentosa, their maculas are relatively normal. The cone photoreceptors have the same packing density as a normal retina. However, when we start to look out into the periphery, we start to see atrophy and death and loss of the photoreceptor layer specifically out into the periphery. And this is because of normal development of the retina having more rods in the periphery compared to the center. Ultimately, though, clinically, what we see then is what's called bone spicules, where we see this pigmentation in the peripheral retina, which we can see in our image on the right. If we look at the fovea, though, the fovea still remains relatively intact in this person with retinitis pigmentosa, even though they have severe bone spicules resulting in severe loss of vision in the periphery. Now, we said that retinitis pigmentosa is about 50 different types of genes or genetic disorders, all affecting rhodopsin. And that means it has a wide variety of different inheritance profiles, including autosomal dominant, autosomal recessive, and X-linked. It doesn't, though, matter what type you terribly have, all ultimately start to result in visual field loss, starting in the periphery and moving towards the fovea. Typically, though, most people with retinitis pigmentosa have relatively intact central vision, resulting in what's called tunnel vision oftentimes. We've known for a long time that this damage in the periphery actually may be causing shifts in people's refractive error. Here's a first study from the 1970s showing a normal population of eyes where we have our typical refractive error averaging around a +2 diopters of hyperopia. Conversely, if we look at retinitis pigmentosa patients, on average they have a 3 to 4 diopter shift into more myopia. And this goes back to the theory of peripheral retina being critically important to normal emmetropization and normal refractive development. If the periphery is damaged, there's a potential error in the signal that can result in changes in refractive error. Now, this ultimately somewhat depends upon the type of inheritance that you have for your retinitis pigmentosa. X-linked, as an example, shows high levels of myopia, whereas most other types on average will show a shift to less hyperopia and more myopia, but not a huge shift like we see in the X-linked and in certain types, mixed types of retinitis pigmentosa. Now, ultimately, this is actually causing errors in our retinal signal to encode the sign of defocus, and that might be why retinitis pigmentosa interrupts normal emmetropization and results in more myopia progression. Thank you. The end.